Exome and genome sequencing have a diagnostic yield of about 30 to 50% depending on the patient's indication, and that's fantastic. But what we're trying to do with RNA sequencing is really push the envelope forward on increasing the diagnostic yield for patients. And so with RNA sequencing, what we're doing is we're looking at variants that exome and genome identify, and then doing RNA sequencing for certain variants to understand if that variant is expected to be associated with disease. And we find that about 4% of patients that have exome and genome sequencing have RNA-seq eligible variants, and about half of those eligible variants end up being reclassified by RNA-seq. This really helps to provide more patients answers, and this is so important in the rare disease setting where every diagnosis matters. We also have our global MAPS test, which is a metabolomic profiling test. And this is done either with exome or genome or other tests as well where a patient is suspected to have a metabolic disorder. And this allows us to either clarify the meaning of variants that we identify by other tests or to potentially identify findings in of itself. What excites me most about where Baylor Genetics is heading is just how quickly developments are happening. Every time I come to one of these large conferences, it's just so clear to me how much work is being done to really understand genetics, genomics, and really trying to tie that into enhancing patient care. And so it really just speaks to, how quickly things are developing.